Ladies and gentlemen, thank you for standing by and welcome to the Antimicrobials and Microbes Indoors, How to Best Promote Indoor Environmental Quality. At this time, all participants are in a listen-only mode. Please be advised that today's conference is being recorded. After the speaker's presentation, there will be a question and answer session. You can type in your questions on the Adobe question and answer box, and we will answer them at the end of the presentation. I would now like to hand the conference over to our first speaker for today, Mr. Jordan Zambrona. Thank you. Please go ahead.
Thank you, Anne. Hello, everyone. I'm Jordan Zambrona, a biologist with the EPA Indoor Environments Division. I'm pleased to host the latest IAQ Science webinar titled Antimicrobials and Microbes Indoors, How Best to Promote Indoor Environmental Quality. Before I introduce our speaker, there are a few points I need to mention. First, this webinar is being recorded and will be made available on the EPA website, viewable on the top left box on your screen at a later date. If you have a question during today's presentation or for during the Q&A session, you can type it in on the left-hand side of your screen at any time. Questions will be read to the speaker for discussion at the end of the presentation. If you have any technical questions or comments, you can submit those inquiries through the Q&A box as well. We look forward to hearing from you. There are two documents available for download on the bottom left portion of your screen. They are a copy of the presentation slides and a PDF copy of the National Academies of Science Microbiomes of the Built Environment. I also want to reiterate that the content of this webinar represents the research and opinion of our speaker and not necessarily those of the U.S. Environmental Protection Agency. With that said, I'd like to introduce our speaker, Dr. Erica Hartmann. She is an environmental microbiologist and assistant professor at Northwestern University. Her interests include the interaction between human-made chemicals and microbes. She began leading studies of antimicrobial chemicals and microbes found in indoor dust at the Biology and Built Environment Center at the University of Oregon, and is currently continuing that work at Northwestern University. So Erica, thank you again for speaking today, and I will hand it over to you to begin our presentation.
Great. Thank you so much, Jordan. Thank you very much for the invitation. It's my pleasure to be here today. There we go. So as the instructions go, I'm happy to take questions at the end of the presentation. Hopefully there will be ample time for Q&A. If not, you are more than welcome to get in touch with me also via social media at Erica M. Hartmann on Twitter or Instagram. So I want to start out just by talking a little bit about what is the indoor environment and who lives there. So very, very broad view. I'm sure we're all well aware that indoor environments have many inhabitants, which include not just us, the humans, but also, for example, our domestic animals, our pets. And we also have maybe some houseplants. Maybe a lot of people are getting into taking care of their houseplants now that we're all stuck indoors. But in addition to those inhabitants that we can see, there are, of course, many inhabitants that we can't see. And if we zoom in on, for example, a dust bunny in your house, you might see all kinds of different life forms or things that were formerly alive, so insects, for example. And if we zoom in even further, we will see, of course, the subject of today's talk, which is our microbial cohabitants. And we see a wide variety of different microbes that are present, not just in dust, but in all kinds of different habitats throughout our built environments, in our homes, in our offices, in our schools, in the air, on surfaces, in water. And these really have a lot of very interesting features, we'll just say. So this has been the focus of a pretty robust research effort, I'll say, that was profiled in this report by the National Academies of Science, and that's one of the documents that's linked in the lower left-hand corner here. I encourage you to check it out. I'll talk a little bit about some of the aspects of it that I think are maybe most relevant to our discussion today or most interesting to me personally. But some of the main questions that were addressed in this report are, what do we know about factors that affect microbial communities? So all of those microbes in all of those various places. What do we know about what makes up those communities and why they are who they are? We also want to know how these communities affect our health and the health of our buildings. So maybe it's structural integrity of the building, but of course we're obviously very concerned about the health of the human and animal and plant inhabitants of our built environments as well. And then how do we get to a future vision in which we understand enough to manage indoor microbes? And I think that's really the key, is that right now, when this report came out, and even today, we don't quite know enough to really say that we can manage indoor microbes, but we definitely want to get there. So what do we know already? One thing that we know about microbes inside of buildings, and especially those microbes that are associated with surfaces, is that they tend to look a lot like human skin microbes. So a lot of the microbes that we find indoors probably came from the human inhabitants themselves, and we see this whether we're looking in kitchens or bathrooms or any type of surface that humans commonly come into contact with. And we also know that that interaction can go in both directions. So we are distributing our microbes, the microbes that we carry around with us, to different surfaces, and we also pick them up while we're passing through an environment. And I think this is maybe intuitive if we had just sort of thought, like, of course you would be inoculated by the microbes that are around you. But it's really interesting to actually see the proof of that and try to quantify that, because your microbial exposures are so important to your developing immune system, for example, for children, and all kinds of other health impacts. And so really understanding the nature of this interaction is really important, and really quantifying that. So, for example, some of the work that I've done has looked at how human-associated a particular spot in a building is. So for any room in a building or any surface within that building, how much does it look like a human microbiome versus how much does it look like another microbiome? And so, for example, in this study, we went around this beautiful old building and collected dust samples from lots of different spaces within that building. And what I'm showing here is a bar chart where everything that's in purple is some sort of human-associated microbe, and everything that's in gray is some sort of environmental or other. And what we found in this particular building is that while we did see a lot of human-associated microbes, most of the microbes were, in fact, environmentally associated. So we think that they came from outside. And in this building, that makes a lot of sense because it's a beautiful old building with all of the air being supplied through these big open windows. Let's see if I can use the pointer here. So we've got these gorgeous windows that are open to let in lots of outdoor air. And let's see. One of the other things that we noticed is that even when the windows were closed, so this photo was actually taken from the inside of the building, we had plants growing in through the gaps in the windows, like between the window sash. And so we sort of thought from this, it's a natural conclusion to think that if these windows aren't keeping out the macroflora, how could they possibly keep out the microflora? So we see that built environments have a lot of contribution of human-associated microbes, but also microbes from the environment, and that one potential factor that can influence whether something is more human-associated or not is where the air is coming from in terms of ventilation. And we've also seen that in previous studies. So in this study here, they looked at offices that either had mechanically supplied air, so the HVAC system, air conditioning, or natural ventilation through open windows. And they saw clear differences between the offices that had mechanical ventilation versus natural ventilation, even though these offices were in the same building off the same corridor. And we saw that again in a separate study that we did of athletic facilities. So here we went into a whole bunch of different athletic facilities throughout the Pacific Northwest. And it looks like it's not showing up perfectly well, but basically we saw more human-associated microbes in athletic facilities that had mechanical ventilation than those that had natural ventilation. And we also saw more human-associated microbes in facilities that were cleaned more often using what we call like wet cleaning products. So it's just sort of an interesting observation. I can't really explain that right now, but there are certainly factors such as ventilation that affect what kinds of microbes are in the built environment around us. So I think that sort of maybe brings everybody up to a very, very coarse understanding of what we know about the indoor microbiome. It comes from either humans or outdoors, and there are a few things that we might be able to do about it, such as changing where the air comes from. But a lot of the microbes indoors really are coming from the indoor inhabitants. So clearly that leaves a lot of information that still needs to be gathered, especially if we want to get to that point where we have a sufficient understanding of the indoor microbiome such that we can actually actively manage our indoor microbial communities, and especially to manage those communities in a way that promotes human health. So again, going back to that National Academy's report, there were 5 major objectives that were identified as really being core to the research agenda going forward. And the one that I really want to focus on here is assessing the effects of exposures on health outcomes. So I'm sure everyone here has been spending a lot of time indoors recently, and maybe you're more aware of it just because of the restrictions on movement. But the fact of the matter is, even before the current pandemic, people spent a ridiculous amount of time indoors. Just upwards of 90 to 99% of your time, if you're living in a developed nation, is spent indoors. And so understanding what types of microbes we're exposed to is really, really important for understanding the development of the human microbiome and really trying to influence those health outcomes. So obviously human health is really important, but we still don't fully understand a lot of the interrelationships between the indoor microbiome and human health, even though this is something that we would love to be able to understand and that people have spent a lot of time and energy studying. So one sort of particular aspect of human health that people have really latched onto is allergies and asthma. So in particular with asthma, looking at exposure to fungi and indoor fungi. And what's really interesting here is you would think that maybe this is pretty straightforward. Maybe where we have lots of indoor fungi, we have lots of asthma because we have lots of allergens. But what we actually see is not quite that straightforward. So several studies have looked into this phenomenon, and I'm just highlighting a couple here, basically showing that where you have a lot of a very specific fungal allergen, then you have children who are more prone to develop asthma. But it's not all fungi. It's really just a few specific fungi that seem to be really contributing to these downstream health impacts. And what's really interesting is that while some exposures, so some microbes, can contribute to really negative downstream impacts like allergies and asthma, there's a lot of evidence that actually is showing that having exposure to a diverse microbial community can actually have quite positive health benefits. So while high exposure to specific allergens when you're young can lead to increase in allergies and asthma, diverse exposures that we don't fully understand what exactly those are, but those diverse exposures can actually have a protective effect and maybe prevent the development of allergies and asthma later in life. So really understanding this relationship between exposures and health outcomes is really important, and understanding exactly what we're being exposed to. So maybe the idea that all microbes are germs and all germs are bad, it's overly simplistic, of course, but it's actually really dangerous because if we, as we'll see later in the presentation, if we take a really strong approach to try to get rid of all of the microbes, we might get rid of some of the quote-unquote good microbes in the process and actually end up having some really, really unwanted, unintended consequences. So I think this is particularly important now in the COVID pandemic because we're obviously really concerned about the spread of infectious disease. And so this graphic here, believe it or not, is not very recent. It's from 2012. I mean, I guess it's kind of recent. But it's showing all of the different pathways that a microbe can take from one person to another. And this sort of is what governs our understanding of how infectious disease is transmitted. So, for example, if we walk through the sort of chronology of controlling the spread of infection from the beginning of the pandemic, we were very concerned about fomites. So this idea that someone would cough and that cough would contain virus particles, and those virus particles would be deposited on surfaces, and then someone else would come touch that surface, pick it up, and then infect themselves. So that's the idea of fomite transmission. And that's what got everyone really, really interested in cleaning all the time, and people are still very interested in cleaning. And that's what got people really, really interested in hand hygiene. And I would say those things are still both very important. However, we're now starting to appreciate that there's also this direct aerosol transmission route where, in fact, the particles in the air don't have to settle on a surface. They can be directly inhaled by someone else, and that constitutes a type of or a path of infection transmission. And both of these pathways happen indoors for a lot of reasons, one of which might be just the fact that we're close to each other when we're indoors. But it also might go back to that question of ventilation. And so there's a lot of really excellent research right now looking at the impact of ventilation, not just on the microbial communities that are indoors, but also in trying to mitigate the spread of infectious disease indoors. That said, it's really important to understand just the fundamentals outside of human health so that we can get a better idea of what the full impact is, because we're not just spreading pathogens. We're shedding microbes around us all the time, whether we're healthy or carrying an infection. And again, some of those microbes may be very important. So, to summarize, the built environment is not sterile, and we as hosts are bringing our microbes in with us all the time. We shed our microbes, the ones that we carry in our gut, in our mouths, on our skin, and et cetera. And we're depositing these microbes on everything all around us. And in order to prevent the spread of infectious disease, for example, we do all kinds of things like I mentioned, practicing proper hygiene. And what's really important in terms of practicing proper hygiene, and hopefully everyone knows this by now, that if you really, really want to wash your hands well, you have to wash your hands for at least 20 seconds. I've been saying this for years, and now I'm really excited that hopefully everyone has gotten the message, and maybe I don't have to say it so much anymore. And hand hygiene is really important. It's really, really important in healthcare settings. It's really important outside of healthcare settings. So no matter what happens, just remember, washing your hands is good for at least 20 seconds, soap and water. Okay, but in addition to washing our hands, we use all kinds of other strategies to prevent the spread of infectious disease. And one of them is to use antibacterial or antimicrobial products. So I think when most people think of something that's antibacterial, we sort of naturally think of hand soap. But there are all kinds of antimicrobial products within our built environments. So one example that I like to give, I'm not sure if Ticonderoga actually still has this in their pencils. They may have changed. But this world's best pencil here, protected by Microban, what that means is that there's actually an antimicrobial embedded in the paint around the pencil. So we can have antimicrobial objects. We can have other antimicrobials in other personal care products, so including toothpaste, shampoos, moisturizers, all kinds of personal care products. And plastics. We love to put antimicrobials in plastics. So this is, for example, a keyboard, but a lot of high-touch plastics, things that you might use in your kitchen, baby toys, all kinds of things, all kinds of objects within our built environment also have antimicrobials embedded into the material itself. And then not just the objects, but carpets, tiles, laminate floors, paints, all kinds of building materials. So not just the products that we use, not just the objects that we use to furnish our built environments, but the very materials that we build out of are often embedded with antimicrobials. So really the built environment, like the presence of antimicrobials in the built environment is really just pervasive, way beyond just your antibacterial hand soaps. And I'm going to talk here for a minute about antibiotics and antimicrobials. One thing I want to make really clear is the way I am about to define these two words is not universally accepted, so it's just for the purpose of this talk. So just for the purpose of this talk, we can consider antibiotics to be medicines for humans or animals. So these are the life-saving drugs that you get, for example, if you have an infection and we need to prevent sepsis, for example. And what's critical about antibiotics is that at least for human health, they're typically given for a very short period of time at a very high dose. So the idea is that you have an infection, we're going to blast it with this antibiotic, and then the antibiotic goes away and the infection goes away and you sort of hopefully go back to normal. This is in contrast to antimicrobials, which are agents used to disinfect surfaces and eliminate bacteria in a way that typically leaves a long-lasting residue. So the idea here isn't a short period of time high dose, but a long period of time leaving some sort of surface residue, some sort of active residue on that surface. So for example, your keyboard is not supposed to be antimicrobial just when you pull it out of the box. It's supposed to stay antimicrobial for, I don't know, as long as you use it. And that's really, really important from a point of view of the microbes because they would experience those two types of exposures very differently. But the types of responses that they use, as we'll see, can actually confer resistance to both. So one critically important idea here is that exposure to antimicrobials can lead to resistance to antibiotics. And that is really, really problematic. So in our effort to sanitize our built environment and to prevent the spread of infection, we introduce all kinds of chemicals into the environment. And these, as I just discussed, can be in personal care products, but they can also be in objects that we use to furnish our built environment and also in the materials that we build the very environment out of. And these chemicals, once they get into the built environment, partition. So they don't always stay where we think they should stay. And this, I should say, is the focus of a whole separate research agenda. I highly encourage you, if you're interested, to check out the Home Chem project, which is a really, really cool resource where a bunch of scientists went into a home and did a bunch of things and then looked at what happened to the chemicals within the home. It's a super cool project. Fascinating, a little scary. But basically, what we know is that we bring all these chemicals into our homes, and they don't necessarily stay, like I said, where we put them. So they can partition out of whatever products they were in originally. Some of them are going to volatilize into the gas phase. Some of them are going to sorb to suspended particles. Eventually, a lot of things end up in settled dust. And so we have these chemicals that are floating all around. They're sort of cycling through our built environments. And so I encourage you, going forward, to think about the built environment not as that static sort of physical structure that surrounds us, but actually as a dynamic chemical and biological system. Because, of course, where we have these chemicals partitioning, we also have microbes, right? We already discussed that we have microbes floating through the air and also on surfaces and also in dust. And everywhere where these microbes and chemicals get together, we have an opportunity for what I'll just call a microbe-chemical interaction. And we'll talk a little bit more about what those potential interactions might be. But the one that we are, I would say the one that I'm most concerned about, at least, is this idea of resistance. So as I mentioned, exposure to antimicrobials, the more a microbe is exposed to antimicrobials, the more chances basically it has to develop resistance. And it can develop resistance in all kinds of different ways. So for example, we can have transformation, let's see, which is what I'm showing over here, where, for example, the microbe encounters a chemical that it doesn't like, and it finds a way to break that chemical down. And in some cases, that's very, very useful. That's what we leverage when we use bioremediation, for example, to clean up oil spills in the ocean. That might be less useful or less desirable in indoor environments where we might want these chemicals to stick around, or we might not want to be exposed to the degradation products. Another way in which antimicrobials can lead to resistance is by favoring resistance mechanisms such as efflux pumps, which is what's being shown here. And these are really concerning because a lot of these efflux pumps are not super specific. And this is where the concern about antimicrobials and antibiotic resistance really comes together, where you say, oh, by exposing this community to antimicrobials, I've actually just encouraged them to become what we call multi-drug resistant. And that is, again, not great. So we can see this already in a couple of case studies. So you might think, oh, well, we have all these antimicrobials, we have all these microbes. Are there actually more resistant microbes in the built environment? And so this study set out to answer that question by looking specifically at Staph aureus, which is a common human commensal, and its closely related cousin, antibiotic-resistant Staph aureus, aka MRSA. And so what they did in this study is they went to homes and they looked at Staph and MRSA in the air inside of homes and compared it to the air just outside of homes. And these are real data that I have animated for your entertainment. But what they found is basically more cultivable MRSA inside of homes than just outside of homes in that air, despite finding roughly the same proportions of Staph overall. And so this suggests to me that there is, in fact, something about the built environment that's, in fact, enriching for antibiotic-resistant organisms. And it may not feel like it right now. It may feel like the only public health crisis is COVID. Unfortunately, that's not the case. So antimicrobial resistant infections are a really, really critical public health challenge. It's only supposed to increase, and so we can project the frightening numbers of deaths due to antimicrobial resistant infections as they increase. And this is, I think, especially frightening because these are diseases that we used to be able to treat, right? Infectious disease was one of the leading causes of death for a very long time, and then only in the last about 100 years, since we've had this growing arsenal of antibiotics, have we really gotten a lot of these infections under control. And now we're starting to lose that control again. We're starting to lose that battle, in part because we are overusing antimicrobials. So I think this is a major, major source of concern, even though it may have fallen out of the sort of forefront of everybody's minds right now. So I'm going to go through two examples of some of the work that my group has done, specifically looking at triclosan, which was commonly found in toothpaste and can also be found in other objects throughout perhaps your home, and also quaternary ammonium compounds like benzalkonium chloride, which are now found widely in antimicrobial hand soaps and also paint. So I'll start with triclosan. So as I mentioned, triclosan is found in toothpaste and other goods. I think it's currently being phased out of toothpaste. It may have already, but for several decades, it was very, very widely used in hand soaps, in toothpaste, and in other products. Thanks to its extremely widespread use, triclosan is ubiquitous in indoor dust. Exposure to triclosan is linked to antibiotic efflux, so this is that main concern about multi-drug resistance. And it's also linked to horizontal transfer of mobile antibiotic resistance genes. So this is just another mechanism by which microbes can develop resistance, and which is really, again, very problematic and difficult to control. So triclosan, as I mentioned, has been used in toothpaste, disinfecting solutions, hand wash. Right now it's been taken out of those, and like I said, I think it's being phased out of toothpaste, but it was very, very widely used for a very long time. From there, as you might expect, it gets washed down the drain and goes to the wastewater treatment plant, which is every environmental engineer's favorite place to study chemicals. It's where we start with a lot of things because it's where a lot of chemicals accumulate. And of course, chemicals don't just accumulate in the wastewater treatment plant. They can then pass through, either sorbed to the solids, which are then maybe applied to land, and so then we find those antimicrobials again on our golf courses and all of the places that we've applied land-applied biosolids. And some of them also come out through the water, so then they can be found in our surface waters and eventually make their way back into our drinking water supply. So of course, none of this is good, but let's back up and stop at the wastewater treatment plant for a second. One of the cool things about wastewater treatment plants is that they are amazing bioreactors where we have this fascinating chemical diversity and also a pretty interesting microbial diversity. And so there have been a few studies that have looked at what happens to the microbes in the wastewater treatment plant when they're exposed to triclosan. So in this particular study, they were looking at a specific antibiotic resistance gene, MEX-B, which is part of one of those multi-drug efflux systems. And what they found is that over time, the abundance of this antibiotic resistance gene is pretty stable. But if we perturb the system with triclosan, so if we expose these microbes now to extra triclosan, in just over 2 weeks, we see over an order of magnitude, so over a 10-fold increase in the abundance of this multi-drug efflux resistance gene. So that's pretty concerning in and of itself. But most of us don't live in a wastewater treatment plant, thankfully. It would probably be a little smelly. But we do live indoors, and not all of the triclosan that we use actually goes down the drain. It turns out that a lot of it gets into the indoor dust because triclosan is what we call a semi-volatile organic chemical. And so when I showed chemicals partitioning throughout the built environment, one thing that happens to triclosan is that it volatilizes and then settles. So there have been a handful of studies that looked for triclosan in indoor dust, and almost every sample that has ever been collected anywhere in the world has had concentrations of triclosan in it that approach what we find in wastewater treatment plants. So this is, again, very concerning. So that, to me, suggested that maybe we should be looking for these types of interactions that we see in wastewater treatment plants also in the built environment. So in this particular study, one of the goals that I had was to first see how much triclosan there was in a building and if that related to the amount of antibiotic resistance genes that we found in this building. And the ultimate goal is to be able to suggest how to control the development of antibiotic resistance in the built environment. So getting back to that idea of having enough information to effectively manage microbial communities indoors so that we can have healthy built environments and promote human health. So for this study, these photos are from when I was still at the University of Oregon. We ran around and collected a bunch of dust samples throughout this mixed-use athletic and educational facility. So this is a giant building that has both sort of gym spaces, classroom spaces, office spaces. And from the dust samples, we extracted DNA to look for antibiotic resistance genes, and we also extracted chemicals to look for triclosan. We also looked for triclocarbon and a handful of other antimicrobials known as parabens. And again, I'm going to focus on triclosan right now. So in our first preliminary study where we were looking just at a single building, we saw higher concentrations of triclosan. We also saw higher abundances of specific antibiotic resistance genes. And what was really interesting is that these antibiotic resistance genes that we identified should not, in theory, confer any advantage against triclosan. So this first antibiotic resistance gene should, in theory, help with exposure to things like a Z-Pak, azithromycin. We also saw efflux pumps, but they're specific to tetracyclines and streptogramins. So we don't understand why there would be this relationship between exposure to triclosan and an increase in resistance to these other antibiotics. But that to me is highly concerning. So we followed up this study and went into more athletic facilities, more buildings than just one. Sorry, this is a little bit blurry. If you really want, I encourage you to go to the publication in M Systems. It's open access. And you can also download these slides to hopefully get a crystal clear version PDF. But essentially what we saw is that in this expanded study, again, in buildings where we saw higher concentrations of triclosan, we tended to see more microbes that had more antibiotic resistance genes. So wider diversity of resistance to different antibiotics. Now, for those of you who are keeping score at home, you may be saying, but Erica, a lot of the microbes indoors are dead and antibiotic resistance genes don't actually always confer resistance to antibiotics, and I totally agree with you. And that is why in our follow-up study, not only did we do DNA sequencing, but we also cultured microbes. So much like the study I showed you a little while ago looking at MRSA, we actually cultured the microbes that we were able to collect in these dust samples. And what we saw, again, sorry, this is blurry, but in buildings where there were higher concentrations of triclosan, we tended to observe microbes that were resistant to more different antibiotics. So we tested their resistance to different antibiotics. And so we're seeing not just a higher diversity of antimicrobial resistance genes, but also actual cultivable, viable microbes that are in fact resistant to a wider variety of antibiotics. So to me, that suggests that we are actively shaping the indoor microbiome. We know this already because we know that we are contributing our microbes, and that through the active ventilation, for example, we are introducing more environmental or human-associated microbes. But in addition, by using antimicrobial products, we may in fact be enriching for antibiotic resistance around us. And this is of course not what we intended to do, but it seems to be a recurring side effect. So we may then say, well, okay, triclosan is maybe not great. And in fact, there's, again, for those of you who are keeping score at home, you know that triclosan is widely fallen out of fashion. So in 2014, Minnesota banned the antimicrobial triclosan from hand soaps. That was followed in 2015 by a ban in the European Union of products that came into contact with food could not have triclosan. Shortly thereafter, Kaiser Permanente, which is one of the largest healthcare providing networks in the United States, banned triclosan as well as several other antimicrobials from all of their hospitals and healthcare facilities. And then finally, sort of bringing up the rear, the FDA banned triclosan and I believe 18 other antibacterials from consumer hand soaps. So if you are getting an antibacterial soap now, it will no longer in the United States contain triclosan. That is not necessarily true everywhere in the world, but you know, small victories. But you can still get antibacterial hand soap in the United States. Now it just contains benzalkonium chloride instead of triclosan. So let's talk a little bit about benzalkonium chloride now. It was specifically excluded from the FDA ban because they said that they should allow for more time for people to collect more data, not because it was deemed safe, but because it was deemed that there wasn't enough information to decide that it wasn't safe or wasn't effective. But one thing that we know already about benzalkonium chloride and quaternary ammonium compounds is that they are ineffective against spores. So spore formation is yet another tool in the vast microbial toolkit for resisting antimicrobials and antibiotics, as well as all kinds of other environmental stressors. In addition, exposure to benzalkonium chloride is also linked to antibiotic transformation and efflux, so these different mechanisms for potential cross-resistance between antimicrobials and antibiotic resistance. So, I wanted to follow up our work on triclosan by also now looking at benzalkonium chloride, since that's, you know, triclosan is totally out. Benzalkonium chloride is the new triclosan. Clearly, we should be studying benzalkonium chloride. And one of the things that I thought was really interesting that came out in about 2016 is this microbicidal paint. So this is, I think, the first and possibly still the only EPA-registered microbicidal paint. It contains benzalkonium chloride, so the same chemicals that we're now using in antibacterial hand soap, you can now buy in paint formula and put on your walls, should you want. It's not, in fact, so unusual to include an antimicrobial in paint, and in fact, most paints that you'll buy contain some sort of antimicrobial to prevent the growth of mold while the paint is in a liquid form in the can. So what we wanted to do is compare what happens to microbes that are exposed to these microbicidal paints compared to something that contains a different antimicrobial that's not necessarily registered as microbicidal, but still some sort of antimicrobial. And then as a sort of additional control group, we found this natural clay paint. There's a lot of interest in reducing the carbon footprint of buildings, and so trying to find something that is less toxic, for example. And this clay paint, to the best of our knowledge, contains no synthetic chemical biocides. And so what we did here is we painted a bunch of different coupons of drywall with these different paints and then exposed microbial communities to these paints. And these microbial communities, I should actually say, were actually the same microbes that we were able to culture from indoor dust. So one thing that I think is really cool about this work is that we're not using E. coli, which is what sort of, it's the sort of the lab rat of microbiology, but we're actually trying to get closer to the authentic, undomesticated built environment microbes here. And one thing that we saw across the board is that bacteria do not like being on dry, barren surfaces. So yes, most of the organisms that get deposited on surfaces in the built environment, in the absence of water, all they do is die. However, there are several microbes that still survive, and what was interesting is that based on what type of paint was on the surface, we saw different community membership in the surviving microbes. So this is on the top here, the clay paints, which contains no added antimicrobials, and we can see it's a little difficult to see here, at least on my screen, but Cochuria rosea is this red guy, is doing pretty well compared to, on the bottom, these are the mold-resistant paints where we see more Bacillus thuringiensis and a little of this yellow guy, the Pantoea. And if we look at this microbicidal paint, what's interesting is, yes, it's microbicidal, but nevertheless, there are still bacteria that survive on those surfaces, and overwhelmingly, we see survival of Bacillus thuringiensis. So we suspected that because we know that Bacillus is a spore former, and we know that benzalkonium chloride is ineffective against spores, we suspected that this Bacillus thuringiensis might be forming spores. So we followed that up, and what you can see here are images of the vegetative and spore form of the bacillus. And then these images were collected from swabs that were taken from our exposed cultures. And we can see, again, the vegetative and spore form of our bacillus. So both of these are present on the surfaces, which is really interesting, and we're now following that up to try to see if, in addition to spore formation, there are other survival strategies that bacillus are using on these surfaces. But again, spore formation is not just a fascinating thing that bacteria do, but also a big issue for human health. Specifically, one of the major antibiotic-resistant infections that we're really concerned about is C. diff. And C. diff is notoriously difficult to get out of different environments because it's a spore former. So it's so hardy in the environment, it's really, really hard to kill once it's in there. So there's a lot of concern that if we're using these antimicrobials, that instead of just getting rid of all of the microbes, we're actually enriching for a different set of microbes than if we didn't use the antimicrobials. And so I think it's really important to think about who do we want to enrich for, because we're never going to be able to get rid of all of them. In addition to concern about antimicrobials and antibiotic resistance, there's now a lot of concern, incidentally, about exposure to quaternary ammonium compounds, just in terms of direct impacts on human health. And in fact, this was the cover of CENN, which is a widely read publication from the American Chemical Society, highlighting some serious safety concerns about these disinfectants, which I don't know if you've noticed, but they're wildly popular right now. So much so that the manufacturers are unable to keep up with demand. So it's really concerning to me that we're using these chemicals basically so widely when they can be impacting the microbes, which can then have human health impacts and also direct human health impacts on us. I'm just going to take a slight detour here to get on my soapbox, since I have one, to talk about the marketing of antimicrobial products. And so one thing that I noticed recently is, well, obviously the market for antimicrobials has skyrocketed because of the pandemic. And it's not just the antimicrobials that you might think about, the Clorox wipes and all of the cleaning products, since we're so concerned about fomites, but also all kinds of other antimicrobial products. So one example that I found here are antimicrobial face masks. And I thought this was particularly interesting because you know, there's a lot of debate about what types of face masks perform the best in terms of filtering out the viruses that are, you know, passing from one side to the other, so trying to block transmission. But I haven't really seen anything that talks about adding an antimicrobial as an extra layer of protection. And I really want to commend this one company for explaining that their antimicrobial textiles, when you include the antimicrobial, it is designed to inhibit the growth and persistence of bacteria and viruses on the textile surface. So to be really, really clear here, it's not saying that if we add an antiviral that we're going to reduce transmission through the mask. It's saying that the mask itself won't harbor quite as many viruses or quite as many bacteria. And that's a really, really important distinction. It's sort of the key distinction between the FDA, which is making judgments based on, you know, does this actually impact human health, and that's why we have clinical trials, and the EPA, which when something is registered with the EPA is saying this contains an antimicrobial, and you can put it there to protect the integrity of the product. So you could say that this is to protect the integrity of the mask itself, or any number of other products that are not specifically for healthcare and are not actually shown to improve the quality of human health. So I think there's a lot of consumer education and advocacy work that really needs to be done in this space. One thing that I think is really interesting, one trend that I think is really promising, I'll say, is this move towards transparency, specifically within buildings and building materials. So there are a whole bunch of different third-party labels that you can now get. Maybe you've heard of like LEED certified buildings. There are a whole bunch of different certifications, both for the buildings themselves and the different types of products that you might use in them. And this is a little bit, I compare this to the early days of organic foods, where there were a whole bunch of different third-party certifiers, and different ones had different criteria. And so, you know, you could see that something was organic or not, but they were all picking different criteria for what they called organic. That's where we're at right now with sort of healthy building materials, is that there are a whole bunch of different labels out there. I don't necessarily know what the criteria of all these different labeling schemes are. I don't know if they exactly align with what I'm looking for in a product, but at least there's a label out there. And labeling is really important because it enables consumers to evaluate different products and to express a choice. So if I know that I want a healthy product, I can opt to buy something that has one of these third-party labels. So that's this reporting enables evaluation. Evaluation enables preferential selection. That is basically market pressure to drive manufacturers to make products that meet the specifications that I want. And in order for the manufacturers to be sort of rewarded for coming up with these cool new products, they need to be able to show off how cool they are. And so then we go back to having labeling, and labeling being a way for them to certify that their product is indeed healthy or sustainable, or whatever it is that we're going for here. So I think that's really promising, and I hope to see a lot more in that area, maybe not just within building materials, but also in, for example, other products that we use in our homes, whether those are personal care products or actual objects, or maybe clothes or objects like decorative objects, that kind of thing. To think about more holistically what all is in these objects. And this is all really important, not just for us, but also for our microbial cohabitants. So I just want to take the last few minutes here to really sing the praises of the microbes because they get an awful lot of bad press, and it's important to acknowledge that they're also really essential for making us and for making the world go round. So microbes make up the vast majority of life. As one of my colleagues likes to say, the biosphere is controlled by microbes and some multicellular contaminants. And this is the sort of most recent drawing of the tree of life, where we can see that everything in purple here is microbial, everything in red here is microbial, most of this green stuff is microbial, and then there's us. So it's really important to acknowledge that the chemicals that we use impact not just us, but this huge amount of diversity that surrounds us. And microbes are really important because without them, we would not have enough oxygen. We would not have enough nitrogen. And we would not be able to digest our food. So these are all really critical ecosystem services that microbes provide, in addition to possibly helping us form really robust and well-functioning immune systems. And yes, there are a few microbes that make us sick, but they're really, really in the minority here. So just to sort of summarize, there are microbes around us all the time, and where we may have thought that using antibacterial or antimicrobial products would get rid of all the microbes and make us healthier, that's in fact probably not true at all. And as an unintended consequence, we may be making the bad guys worse, essentially, by enriching for antibiotic resistance. And in addition, all of these antimicrobials that we use can not just affect the microbes themselves, but they can have some really negative direct human health impacts, such as acute toxicity, endocrine disruption. They can cause cancer and birth defects. And so it's really important to use cleaning products judiciously and in a well-ventilated area, I'll say. We'll just start with that. But I think that's a topic for a whole nother talk on indoor environmental quality. So the question that really keeps me up at night is this overarching question of how microbe-chemical interactions affect the indoor exposome, and how does our choice of the types of products that we buy and the types of chemistries that we use in those products ultimately affect the microbial communities around us and our own health. And this is I think this is the take-home message that I'm workshopping here, so let me know how you feel about it. But the analogy is that in the same way that helmets have changed but not eliminated head injuries, antimicrobials are poised to change but not eliminate infection. So this is one tool that we should be using in our arsenal to protect human health and prevent the spread of infection, but we should use it carefully and not think about it as the silver bullet, as it were. So with that, I would like to thank my lab group, all of my wonderful students and postdocs who did all of the work that I shared, as well as so many more interesting studies, all of the funders who have supported my work and the work of others in this area, especially the microbiology of the built environment and chemistry of the built environment programs through the Alfred P. Sloan Foundation. And I will take any questions.
Great. Thank you very much, Erica. That was a fantastic presentation. And despite our bit of a late start, we actually ended the presentation right on time. And so with that, we can begin with some of the Q&A. And so the first question we have here is, what do we know about which taxa might have beneficial health impacts?
So not enough. As a good scientist, I will say we always need to know more. We always need to do more research. I think there are a few taxa that have been identified that are associated with soil that can increase happiness in gardeners. And there may be some taxa that are specifically associated with dogs that can promote maybe some sort of specific positive health outcome. But I think in general, we don't really know. There's a lot of thought that it might be some sort of diversity impacted thing. So it's not just one specific taxon, but you have to be exposed to a certain amount of diversity. But we don't fully understand when and how that exposure needs to take place. So let's keep studying it.
Great. Yeah, that's. I think that's an important point to get across. That there's very, very much yet to be discovered about what has beneficial health impacts. And along that same lines of that question, what kinds of exposures to microbes might be beneficial for young children's lifelong health? So you kind of mentioned having a dog, but kind of what other activities or exposures has the research shown that might have beneficial impact?
Yeah, I think for whatever reason, having a dog is key. Most of the studies that I'm aware of have really focused on the difference between households that either have dogs or are sort of farming or farming adjacent households, and specifically farms that raise livestock. So it seems like there might be something to do with animal-associated microbiomes and maybe some of the microbes that are associated with those animals, although that's not entirely clear. And for whatever reason, the protective health effects don't hold for cats. So sorry for all the cat people out there.
Right. And you mentioned home chem during your presentation, and I thought, and along those lines, you mentioned kind of the differential partitioning of the antimicrobials to different surfaces in the household depending on kind of their chemical properties. Is that something that you know is actively being looked at as a part of home chem? Was there actually data taken specifically looking at kind of antimicrobial chemicals and their chemical components?
So yes, and we need to do more. So I know that people involved in that project and in the chemistry of indoor environments program have looked at impacts on indoor air quality when cleaning with bleach, which is of course a really fantastic and wonderful cleaning product, but one that should be used carefully in well-ventilated areas. So looking at what kinds of secondary chemical products are formed in the air when you use bleach. And I know that they looked at volatilization of other components of cleaning products like the pine scents, those types of fragrances and other components of the cleaning products. But I don't believe they looked specifically at the added antimicrobial chemicals, so the specifics of the triclosan or benzalkonium chloride or other specific antimicrobials like that. So I think there might already be a publication on the bleach impacts on indoor air quality. If not, there's definitely a blog post. And there's definitely some data on the fragrances from cleaning products, but not, to my knowledge, of the specific antimicrobial chemicals.
Okay, great. So perhaps a topic of future research for sure.
Yeah, I hope so.
All right, and kind of taking things to a broader scope. So what do we know about the global trends of antimicrobial use? Are there more countries, more regions of the world that use much more of it than others? And if so, just kind of what may those regions be?
Ooh, I don't fully know. I know that we can look at where antimicrobial chemicals are being produced. I think a lot of production is pretty highly localized, and I can't remember off the top of my head. I think maybe to Germany and a few other places. I know there's an enormous amount of production of benzalkonium chloride and other quaternary ammonium compounds right now. I'm not aware of the sort of global trends. I think a lot of it depends on local markets and sort of local consumer preferences as well as local regulations and restrictions.
All right. We have another question here. Let's see. So what do we know about the effectiveness of cleaning without antimicrobials, so say with soap and water as compared to cleaning with antimicrobials?
Yeah, that's that is the key question, isn't it? We do know from studies on hand washing that an added antimicrobial in the soap does not make washing your hands any more effective. So wash your hands with regular soap and water for 20 seconds. You don't need to use antibacterial hand soap. The detergent in soap and the way we use soap itself is antibacterial enough. In terms of for other products, like throughout how you might clean an industrial kitchen, for example, I think there have been a handful of studies that have looked at specific individual products, like maybe floor tiles, but I don't think there's a general consensus of like which products work best, and you know, I think, so this is getting a little off topic, but I think one of the difficulties there is actually figuring out what metric to use. So I mentioned that with our study of paints, one of the things that we did is we took some of the isolates that we were able to culture from indoor dust and exposed this mock community of microbes to that surface. And that's not typically what's done to evaluate effectiveness. To evaluate effectiveness of antimicrobial products, typically what we're doing is we're taking one type strain, one very well characterized pathogen, and exposing that. And what happens in vitro in the lab during that standardized test is not necessarily reflective of what really happens, out in the wild in your actual buildings. In part because people don't always use products exactly as suggested, and in part because the microbes that we're using versus the microbes that are naturally present in these environments might be very different, and they might have very different survival strategies. So I think it's really important not just to think about, what is and isn't effective, but also to think about how we evaluate efficacy, especially as we develop a more holistic picture of the types of microbes and the types of microbial diversity that's present in these different environments. In the interim, I highly recommend that you use plain soap and water.
Great. So if we wanted to start buying products that do not have antimicrobials embedded in them, are there any specific products that you recommend we start with? Is there a consistent way to identify these products? And that kind of ties in with your labeling towards the end of your presentation.
Yeah, so Jordan or maybe somebody at EPA can correct me if I'm wrong here. But one of the things that drives me a little up a wall is that it's very difficult to get product like ingredient declarations. So anything that's regulated by the FDA, so your soaps and your toothpaste, have to list all of the ingredients. And they'll say the active ingredient is, whatever, zinc or triclosan or whatever it is. And then they'll list, salts and detergents and emulsifiers and all of those other fun things that make our products behave the way we expect them to or have a certain color. The same is not true, for example, for my desk. Somebody might say, oh yeah, it's a solid oak wood desk. Sure, but it's stained with something, it's treated with something. There are all kinds of chemicals that might have been used in this desk, which incidentally, I don't even know if it's actually solid oak, I just made that up. And the fact of the matter is I don't know what's in it. And I don't know what's in my t-shirt, and I don't know what's in many of the other products that I have in my home, in the same way that I have access to that information for a personal care product. So one thing that I would really, really like to see going forward is increased transparency in the materials themselves, and even being able to find out, like, oh, okay, so what really is in these different products. In terms of something more straightforward, is it antimicrobial or not? I would say that's still kind of tricky, because my understanding is that things that are advertised as having an antimicrobial, so if it has the antimicrobial label on it, must contain an antimicrobial, but they don't have to tell you what that antimicrobial is. But things that are not advertised as antimicrobial don't necessarily have to have a label. So I've run into that problem a few times recently, oddly enough, buying shoes, because a lot of manufacturers like to put antimicrobials into the footbed to keep the shoes from smelling, because my feet smell, like, you know, it's natural. So you can see there are some shoes that are advertised as having an antimicrobial footbed. Some of them will tell you that they use, for example, silver, but they probably won't tell you what specific type of silver antimicrobial they use. And the ones that aren't advertised as such, you don't know that they're not. Sorry, that was a lot of negatives. So it's unfortunately tricky, and I think this trend towards transparency, and this is really where like buying things that where the manufacturer has gone out of the way to be third-party certified, at least shows some sort of good intention on the part of the manufacturer to make products that adhere to these standards.
Great, thank you. So kind of turning to another topic, or a tangential topic, I would say, is can microbial communities release VOCs? And what impact do those VOCs have in the indoor environment?
Ooh, yes. So, and there's Pavel Mishtal, who is now, I believe, at University of Texas, Austin, has a really cool research program in this area looking specifically at fungi and microbial VOCs. So absolutely, and I would say anytime you smell like must, that musty smell, that is microbial VOCs, and there are also probably many more that we can't smell. I don't know what specifically the impacts of these VOCs are. I think because a lot of them, unless you have like really bad water damage, for example, a lot of them are very, very weakly concentrated. And so it's been technically very challenging to measure some of these compounds, and technically very challenging to link, for example, specific compounds with specific microbes. Also because we understand so little about what types of functions these microbes might actually be performing in the environment. But hopefully, as we sort of push the limits of our developing methodologies and get better, more sensitive technologies, hopefully we'll be able to answer that question a little more concretely.
Great, great. The next question is kind of in regards to using UV lights in HVAC systems. What do you know about the impact of that on the indoor microbiome?
Oh, I am going to refer you to many of my esteemed colleagues who specifically study HVAC systems. So Shelley Miller, for example, or Brendan Bohr. So Shelley Miller is at University of Colorado Boulder, and Brendan Bohr is at Purdue, and they both specialize in HVAC systems and the microbes that grow within those HVAC systems.
Great. Okay. What are common disinfectants used in schools that we see sprayed from elongated wands? And should we be concerned that too much disinfecting on surfaces could be a problem within schools?
I'm not sure what's commonly used in schools, but I do know that excessive use of disinfectants is probably not a good thing, and we've seen some really, really egregious overuse of disinfectants in I think it was ICE detention centers a few months ago, where they were just using like really, really way too concentrated solutions of, I think it was quaternary ammonium compounds, and using them way too often, and you can actually start to get acute toxicity, so eye irritation, respiratory irritation. Hopefully schools are not using disinfectants to that extreme. But there is a lot of there's a lot of fear right now, and I think one of the ways that we're trying to control that fear is by doing the things that we can, which is cleaning. And I think here we should really talk with like a social psychologist or something to really get a handle on this. But it's really important to balance our chemical exposures with our microbial exposures. And so I would like to see more research in this area, and in the meantime, exercise restraint.
Absolutely. So kind of, we spent this presentation really focused on bacteria within the indoor. This question is kind of asking, do we see similar links between, say, azole-resistant fungi and antimicrobials that we see that antibiotic resistance and antimicrobial exposures?
Yeah, it's a great question, and there are a few, unfortunately, highly resistant healthcare-acquired fungal infections that are really sort of concerning emerging right now. And that's absolutely something that I would like to look into. And that's, I should say, healthcare-acquired infections are often at the sort of forefront of these types of studies because they're very well documented. But I think it's also critical to think about not just what's present in our hospitals, but what's present in our homes, especially since we're using a lot of the same chemicals in our hospitals and in our homes. And so maybe that's not something that we should be doing, and maybe we should be concerned about these types of infections, not just in hospitals and in homes. I think there's certainly some evidence that we're seeing an increase of resistant fungal infections, at least in hospitals. But I think that's maybe an emerging area of research.
Okay, great. Let's see here. What are your thoughts on kind of the proactive use of microbes when applied in indoor environments? Do you think that we know enough about them that is a recommended course of action?
I think it is a very interesting course of action. I actually am part of a study right now where we're looking at probiotic cleaning products. And that project has unfortunately been a little bit derailed while our laboratory was shut due to COVID, but hopefully within the next few months to a year, I'll have some results to share on that, because I do think it's a really, really interesting and promising new strategy, but I think there are a lot of technical issues that we need to overcome to really understand what we're doing. Again, at this point, we don't fully understand the microbial ecology of the built environment well enough to effectively manage it. This could be a very useful way to contribute to that, but I would like to see more studies and really understanding what the full impacts are. And part of that goes back to how do we evaluate efficacy. So there have been a few studies looking at the use of probiotic cleaners in hospitals because, again, this is a place where we're really concerned about infection. And there are some hospitals where they've been able to show a decrease, like a really, really pronounced decrease in hospital-acquired infection following the use of the probiotic cleaning products. But other hospitals have not been able to reproduce that phenomenon. And I think one of the limitations is that we don't fully understand the mechanism by which these probiotic cleaners work or don't work. And so it's hard to understand why they might work in some situations but not in others.
Okay, great. So again, the results are mixed, and we just need to dig further into the research before we can have conclusions about that kind of use of microbes indoors.
Yeah.
Right. Well, I believe that we've come to the end of the questions that we have. And so I would just like to wrap things up by saying thank you for taking the time to present this very interesting research, Dr. Hartmann, and we really appreciate you coming and discussing this with us today.
Well, thank you so much for inviting me. Thanks to everyone who came. Thanks for your attention. And yeah, stay tuned because there's so much more exciting research coming out. I think this is a really important area. So hopefully, hopefully there will be much more in the coming years.
All right. I think, operator, I think we will bring the webinar to a close.
Thank you. Ladies and gentlemen, this concludes today's conference call. Thank you for participating. You may now disconnect.